Today, we are going to learn about an interesting aspect of ecology in terms of biotic community. Let us first start with the understanding of prescribed meaning of community. In nature, different kinds of organisms are found in association with each other. In general terms, a group of several species living together with mutual tolerance referred to as adjustment and beneficial interactions in a natural area is known as a community. A forest, grassland, desert or a pond are the examples of natural communities. Precisely speaking, all the organisms of community live together, share same habitat, influence each other's life directly or indirectly by potential interactions and have reached a survival level within a given radiant energy. Hence, in ecology, community or biokoinosis is an assemblage of populations of two or more different species occupying the prescribed area of habitat in a particular time. By definition, a biotic community must include only living entities of the area of an ecosystem. The concept of community is by no means so recent, and it may be traced back to the time of Theophrastus, who has recognized the existence of plant communities in the different environmental areas. Community ecology, also called Syncology, deals with the study of interactions between species in communities on many spatial and temporal scales, including the characteristic, composition, structure, origin and development, as well as different characters used to describe a biotic community. According to Krebs in 1985, a community possesses the attributes like co-occurrence of species, reoccurrence of groups of the same species and homeostasis or self-regulation. Community is a larger unit than the population. It achieves many characteristics that are not found in its constituents, that is, the organisms and the populations. Size of the communities may vary from small patch of land or water body to extensive forest. Communities differ from place to place and at the same place at different times. Minor communities are greatly influenced by inputs from adjacent communities, while major communities are relatively more independent and self-sufficient of their habitat. In terms of the approach of scientists to community studies, the zoologists are mainly concerned with the functional relationship with a community, while the botanists are mainly focusing on the community structure and the changes occurring in it with time and space. Let us now move on to the very brief classification of different types of communities. Ecologists classify the communities from different viewpoints on the basis of general growth, composition, shape, etc. of vegetation and organisms associated with them in terms of forest, desert, grasslands, tundra and so on. Likewise, communities may be divided as hydrophytic in predominantly aquatic habitats, mesophytic in moderately moist soils and xerophytic in arid or dry conditions. Similarly, communities growing in condition of abundant light are called heliophytic and those growing in shade are called cyophytic. Clements in 1916 noticed that plant communities are not always the same at any place, and he classified the communities on two parallel lines. One in the process of change, which is called seral communities, and the others are called stable or climax communities. Further, the global community comprises of all the plants and animals in the world and is further divided into continental communities and oceanic communities. Now, let us have an overview of various characteristics of biotic community. Like a population, a stable and self-sufficient community too has certain specific characteristics. The characteristics with reference to community level of organization include species diversity. Each community is formed of a number of different populations comprising of all the organisms like plants, animals, microbes, which differ taxonomically from each other. Species diversity depends upon size of the area, diversity of habitats in that area, soil type, altitude, etc. There are two levels of species diversity: regional diversity of whole nations or part of continents within which many different communities exist, and local diversity in a given nation where different communities exist at different latitude. Growth form and structure. Generally, the community is described in terms of major growth forms as trees, shrubs, herbs, mosses, etc., which determine the structural pattern of a community. In each growth form, as in trees, there may be different kind of plants as broadleaf trees, evergreen trees, etc. According to the mode of arrangement of various growth forms, the community shows either horizontal layering called zonation or vertical layering called stratification. Habitat. Biotic community occupies particular area with specific physical environment known as habitat. It acts as a limiting factor and regulates the population size of various species within the community. Dominance. In each community, all the species are not equally important. There are relatively only a few of these which determine the nature of the community. These few species exert a major controlling influence on the community by virtue of their size, numbers or activities. Such species are known as dominants. Succession. Each community has its own developmental history. It develops as a result of directional change in it with time, which is referred to as succession. Trophic structure or self-sufficiency. Nutritionally, each community formed by a group of autotrophic plants as well as heterotrophic animals exist as a self-sufficient, perfectly balanced assemblage of organisms found in series. The remains and dead bodies of the producers and consumers are decomposed by the decomposers like bacteria and fungi, which help in the recycling of materials to maintain self-sufficiency of biotic community. Each community has its own composition, structure, origin and developmental history. Let us have a look at composition of biotic community. In terms of composition, communities may be large or small. Large communities extend over areas of several thousands of square kilometers such as forest, while communities such as deserts are comparatively smaller with dimensions in hundreds of kilometers, and still others such as meadows, rivers, ponds, etc. occupy a more restricted area. Extremely small sizes communities are the groups of microorganisms in microhabitats such as leaf surface, litter, soil, etc. In each community, there are diverse species. All the species are not equally important, but a few overtopping species control the growth of other species of the community by their density and modification of environment, thus forming a sort of characteristic nucleus in the community. These species are called ecological dominants. Generally, most communities possess only a single dominant species and in such case the community is called by the name of dominant species is, for example, spruce forest community. In other communities, there may be more than one dominant, as in oak hickory forest community. Now, let us see how we can describe the structure of a biotic community. Besides composition and dominance, the communities exhibit a structure of recognizable pattern in the spatial arrangement of their members. Organisms are distributed unequally throughout the biotic community, resulting in stratification. Stratification forms one of the outstanding general principles of community structure. The community may show horizontal or vertical stratification and is different in aquatic and terrestrial habitats. Let us start with the horizontal stratification first. A community may be divided into sub-communities which are units of homogeneous life forms and ecological relations. These horizontal divisions constitutes the zonation in the community. In shallow ponds, the zonation is very little. However, in deep ponds and lakes, there may be recognized three zones, viz. Littoral zone, limnatic zone and profundal zone. In each zone, organisms differ from each other. Now, let us also understand the structure of biotic community in vertical stratification by taking certain examples. Vertical gradients in the environmental factors such as the availability of sunlight, the temperature and so on bring about a recognizable stratification in water bodies, particularly in marine ecosystem. The oceanic region, that is the region of open sea beyond continental shelf, comprises of two zones. First, that is bathyal zone, that is region of continental slope and rise, and the second, that is abyssal region, that is area of ocean deeps. In terms of life penetration, the ocean region is vertically also divided into an upper thin euphotic zone, that is the light compensation zone, and a vastly thicker permanently darker zone, the aphotic zone, which includes bathyal and abyssal zone. The oceanic region also includes two more vertical zones, the benthic, that is bottom, and the pelagic, that is whole body of water. The community of pelagic zone includes phytoplanktons, which comprise of the producers such as diatoms, dinoflagellates, flagellates, green algae, brown algae such as sargassum, and zooplanktons means the consumers such as foraminifera, radiolarian, annelid worms, swimming snails, jellyfishes, ctenophores, etc. Nectons, which are free swimming marine animals and essentially independent of water movements. Nectons are consumers such as cephalopods like sepia, bony fishes, sharks, sea turtles, whales, seals and seabirds. Minute organisms that float or swim on the surface water are called neustons. Benthos are bottom dwellers of benthic environment. They are sessile forms such as chitons, oysters, sponges, corals, anemones, crabs, lobsters and flatfishes. Vertical stratification in communities is also observed in terrestrial ecosystem. For example, in a grassland community, the euphobic zone consists of the herbaceous stratum consisting of grasses and other herbs as producers and rodents, cattle and insects as consumers. The subterranean stratum consists of different layers of soil containing litter, humus, mineral fraction of soil, plant roots, rhizomes and organisms such as bacteria, fungi, protozoa, microarthropods, insect larvae, pupae, oligochaetes and mollusks. In forest community, however, the vertical stratification is most complicated and includes 5 subdivisions: subterranean subdivision, forest floor, herbaceous vegetation, shrubs and trees. In some tropical rainforest, 3 more subdivisions exist in the forest floor, such as leaf litter layer called L layer, partially decomposed leaf litter layer called F layer and a humus layer. Thus, based upon the light and relative humidity requirements, we find stratification in above ground parts. Ecological potential of a zone or strata determines the distribution of community. Fenton in the year 1947 has classified these communities as microbiota, which include bacteria, algae, fungi, protozoans, rotifers, nematodes and mites, as well as mesobiota, which include many nematodes, centipedes, millipedes, spiders, insects, etc., and macrobiota, which include plant roots, earthworms, toads, lizards, snakes, rodents and large mammals including man. He also classified these communities according to their feeding habits, such as chemophagous, actophagous, anthophagous, predators and shelterers. Another aspect of biotic community is its origin and development. Communities are never stable, but dynamic, changing more or less regularly over time and space. The occurrence of relatively definite sequence of communities over a period of time in the same area is known as ecological succession. Let us briefly outline the process of succession. In this case, first of all, in a barren area, there reach the seeds and propagules of the species, which is known as migration. These seeds, after germination, developed into seedlings, which then develop into adults. But only a few of these survive and are capable of successful growth, and this process of seedling establishment and successful growth is called echesis. As a result of migration and subsequent echesis, species colonize the new areas referred to as colonization, which ultimately leads to development of climax community. Now, let us gain a detailed knowledge about various characters used to describe biotic community. To study the details of biotic community, number of characters are used for the purpose of expressing the structure of plant community. These are broadly classified into 2 major categories: analytical characters and synthetic characters. Let us learn about analytical characters first. These are generally expressed in terms of 5-point scale and divided into 2 types: quantitative characters, which are expressed in quantitative terms, and qualitative characters, which are expressed only in qualitative way, although finally grouped under 5-point scale. Let us study about first type of analytical character as quantitative characters in detail. Frequency. Frequency is the number of sampling units expressed as percentage in which a particular species occurs. Various species of the community are recorded by different phytosociological methods by taking any sampling unit like quadrate, transect and point. Frequency of each species is calculated using the formula: frequency in percentage is equal to the number of sampling units in which the species occurred divided by the total number of sampling units studied and multiplied by 100. After determining the percent frequency of each species, they are distributed among Ronkier's 5 frequency classes as A 0 to 20%, B 21 to 40%, C 41 to 60%, D 61 to 80% and E 81 to 100% depending upon their frequency values. Density. Frequency does not give correct idea of the distribution of any species unless it is correlated with other characters such as density, etc. Density represents the numerical strength of a species in the community. The number of individual of species per unit area is its density. Density gives an idea of degree of competition. It is calculated by the formula density is equal to total number of individuals of the species in all the sampling units divided by total number of sampling units studied. Abundance. It is the number of individuals of any species per sampling unit of occurrence. It is calculated by the formula abundance is equal to total number of individuals of the species in all the sampling units divided by number of sampling units in which the species occurred. But abundance of quantitative characters gives little idea of the distribution of the species. Cover and basal area. Quantitative values of frequency, density, etc. do not give correct information on the distribution of a species due to different growth forms. So, such values are to be supplemented with other character like cover and basal area. Cover term is generally used for above ground parts such as leaves, stems and inflorescence which cover a certain area. If this area is demarcated by vertical projections, the area of the ground covered by the plant canopy is called foliage cover or herbage cover or canopy cover. Basal area refers to the ground actually penetrated by the stem and is readily seen when the leaves and stems are clipped at the ground surface. Now, let us learn the second type of analytical characters as qualitative characters. Physiognomy. It is the general appearance of vegetation as determined by the growth form of dominant species. On basis of appearance, it may be a grassland, desert, etc. Phenology. Phenology is the scientific study of seasonal change, that is, the periodic phenomena of organisms in relation to their climate. A study of the date and time of different events is phenology. In the other words, phenology is the calendar of the events in the life history of the plant. These events are represented by phenograms. Stratification. Stratification is the way in which plants of different species are arranged in different vertical layers in order to make full use of the available physical and physiological requirements. Abundance. Although related with density, abundance may not be expressed generally in quantitative terms. Organisms, particularly plants, are not found uniformly distributed in an area. They are found in smaller patches or groups, differing in number at each place. Abundance is divided in 5 arbitrary groups depending upon the number of plants. The groups are very rare, rare, common, frequent and very much frequent. Sociability. It denotes the proximity of plants to one another. Species with same density may differ in sociability values. Thus, sociability expresses the degree of association between species. Braun Blanquet in the year 1932 used only 5 sociability groups to classify plants such as S1, that is plants, especially stems, found quite separately from each other, thus growing singly. S2, represents a group of 4 to 6 plants at one place. S3 denotes many smaller scattered groups at one place. S4 indicates several bigger groups of many plants at one place, and S5 is a large group occupying larger area. Vitality. This is the capacity of normal growth and reproduction which are important for successful survival of a species. The vitality depends upon weight of species. In plants, stem height, root length, leaf area, leaf number, number and weight of flowers, fruits, seeds, etc. determine the vitality. On this basis, plants are grouped into different vitality groups. Misra and Puri in the year 1954 gave 3 groups as well-developed plants completing their life cycles regularly, producing viable seeds, plants reproducing vegetatively, and ephemerals short-lived. Daubenmeier in 1968 gave vitality group as V1 representing plants whose seedlings die, V2 denotes seedlings grow but unable to reproduce, V3 is a reproduce only vegetatively, V4 denotes reproduce sexually but uncommon, and V5 that is which reproduce sexually and grow regularly. Life form or growth form. A life form is the sum of adaptation of the plant to climate. On the basis of the position of the perinating birds on plants and the degree of their protection during adverse conditions, Ronquier classified plants into 5 broad life form categories. Phanerophytes. Their birds are naked or covered with scale and are situated high up on the plant. Depending upon the size, they may be megaphanerophytes that is over 30 meters high, mesophanerophytes that is 8 to 30 meters high, microphanerophyte that is 2 to 8 meters high, as well as nanophanerophytes that is under 2 meters. These life forms include trees, shrubs and climbers, generally common in tropical climates. Chamephytes. Their birds are situated close to the ground surface. They are commonly found in high altitudes and latitudes. For example, Trifolium repens. Hemicryptophytes. Their birds are hidden under soil surface. Examples include most of the biennial and perennial herbs of cold temperate zone. Cryptophytes. Their birds are completely hidden in the soil as bulbs and rhizomes. Most of them are found in arid zones. Cryptophytes also include halophytes, that is marshy plants with rhizomes under the soil, and geophytes, that is terrestrial plants with underground rhizomes or tubers. Therophytes. These are seasonal plants completing their life cycle in a single favorable season and remain dormant throughout the rest unfavorable period of year in the form of seeds. They are common in deserts. Now, let us briefly outline synthetic characters. For comparing the vegetation of different areas, community comparison needs the calculation of their synthetic characters. These are actually computed from qualitative and quantitative analytical characters. They are determined in terms of presence and constants. It expresses the extent of occurrence of the individuals of a particular species in the community. The species on the basis of its percentage frequency may belong to 5 different presence classes. Rare, which present in 1 to 20% of sampling units, seldom present, which is present in 21 to 40% of the sampling unit, often present, which is present in 41 to 60% of sampling units, mostly present, that is present in 61 to 80% of sampling units, and constantly present, that is present in 81 to 100% of the sampling units. Fidelity. This is the degree with which a species is restricted in the distribution to one kind of community. Such species are sometimes known as indicators. The species have been grouped into 5 fidelity classes as first formulated by Braun-Blanquet. Fidelity 1. Plants appearing accidentally, that is strangers. Fidelity 2. Indifferent plants may occur in any community, that is indifferents. Fidelity 3. Plants occurring in many communities but predominant in one, that is preference. Fidelity 4. Plants specially present in one community but may occasionally occur in other communities as well, that is selectives. And Fidelity 5. Plants occur only in one particular community and not in others, that is exclusives. Dominance. Here the dominance is expressed in synthetic forms. On the basis of density, frequency and dominance values, there has been proposed the idea of important value index represented by IVI. For IVI values of relative density, relative frequency and relative dominance, that is cover bases, are obtained by different formulas like relative density, that is equal to density of the species into 100 divided by total density of all the species. Relative frequency, that is frequency of the species multiplied by 100 divided by the total frequency of all the species. Relative dominance or cover, that is equal to dominance or cover of the species multiplied by 100 divided by total dominance or cover of all the species. Now for IVI, 3 values are added. IVI values of different species are then arranged in decreasing order. This index is used to determine the overall importance of each species in the community structure. It provides the idea of the sociological structure of a species in its totality in the community. Other synthetic characters. These include interspecific association and association index, index of similarity, dominance index, species diversity and diversity index, etc. Thus, we can say that characters description play very significant role in determination of structure of any biotic community of an ecosystem.
In this particular lecture of environmental biology, we have gained an idea of very important aspect of community ecology in terms of biotic community. Initially, we have seen the precise meaning of biotic community and then its brief classification. During this lecture, we have also seen specific characteristics of biotic community. Not only this, we have also studied composition, structure and development of biotic community with certain examples. In this particular part, we have mainly focused on to the characters of plant community in terms of analytical and synthetic characters to describe structure of any community in an ecosystem.